So Kushbo asked me to focus this talk mostly on the kind of the big pictures of layer-dependent fMRI, if I didn't misunderstood, like kind of what are the underlying principles? Well, what is it good for? What do people like use it for? So this is what I will try to present today, though note that I'm a technical person. I'm a physicist. Specifically, I'm a physicist looking into acquisition methods for high resolution imaging. And as exciting as it is to do so, like I think it's already quite informative to do fMRI at conventional resolution. So conventional fMRI at lower field strength has already a lot to offer to understand kind of the function of the big brain structures in the brain, like the ones, for example, that you see here, like the part that's responsible for vision, moving the body, listening, or these uniquely human abilities like language. However, as exciting as it is to see the brain in action like that, sometimes it's also a bit frustrating how little we learn about the brain by looking at these kind of big structures and these kind of fuzzy blobs that you see here. Sometimes, jokingly maybe, I compare it as trying to understand how the brain works by looking at these fMRI blobs is somewhat comparable as trying to understand how a truck engine works by looking at it with an infrared camera. You kind of roughly see the big structures that are involved, right? The front seems to be consuming a lot of energy and heats up, so that must be responsible for driving. But we don't really learn a lot about kind of the densely packed microcircuitry, the mechanics underlying it, right? So at high field strength and high spatial resolutions, we can not only distinguish between like voxels that have tissue types of gray matter, white matter, and CSF, but also we can start to resolve finer structures within the gray matter, aka the cortical layers. And these different layers, they have different neurons that do different things, different kind of processing. So going from these kind of low spatial resolutions of 3 by 3 by 3 to sub-millimeter resolutions accessible at ultra high field strength allows us to distinguish those different neural populations better, allowing us a new kind of insight into the mechanics of the underlying processes. So we not only learn if a brain area is involved in a certain task, but also more information about where this activity is coming from, right? How it's following the different processing step, how it's modulated from other brain areas until we have output that's sent to further areas with respect to feedback and so on and so forth. So with layer-dependent high-resolution fMRI, we not only learn which brain areas are connected with each other and which of those connections are stronger than others, but importantly, we learn something about the causality, the directionality of this information flow, which can be quite exciting for neuroscience, for example. Though to get to these high resolutions is a huge challenge, and the field has come a long way, and there's still a long way to go. And I would like to exemplify this with this little metaphor of astronomy. So if we look at this beautiful night sky and you have a very sensitive camera, you can see that there are these kind of spots, these clouds. And if you zoom in, there's a lot of structure in there. And if you zoom in further, you see that there are even like finer scale structures further and further and further until you ultimately hit the resolution limit of earthbound telescopes like La Silla in Chile, right? And then with a huge hardware boost going to space, you see a way more structure, right? You see even some dynamics within those clouds. And with further improvements, ultimately, you see like even more structure, right? You see even kind of how these clouds build up by just looking at this image. There's a lot of details there. So the resolution is key. And I like this metaphor because this is just about the time scale of fMRI development too. Like the biggest leap forwards was the invention of it in the early 90s, where the resolution was quite coarse actually. But you could see non-invasively that you could distinguish between different brain systems, like the somatosensory areas compared to other areas. Now, with a lot of hardware improvements, more than 2 decades later, we have 2 orders of magnitude and even more in resolution gain down to 1 millimeter resolutions, and where we start to see kind of that we separate the sensory areas from the motor areas, which are very close together. Now, the Moore's law of fMRI resolution continues, which gives us about an order of magnitude every decade. So a decade later yet, we are down at about 0.1 microliter. where we also see kind of distributions within the gray matter, which is quite exciting because there is a lot of information down there. And namely, you can see the activity patterns here of voxels kind of forming a line along the cortical ribbon, actually two stripes over there, which we would like to interpret as the different layers. And then there are also these kind of pillars, columnar-like structures that not many people have seen yet. So we don't really know what they are for, or if they are more vascular artifacts. So I will talk about the layers mostly. The thing I want to show you on this image, though, is that there's a lot of structure, a lot of fine scale information in the functional responses. If you just look at the EPI, the underlay, the structural contrast, the anatomical contrast, there's not so much you gain from it, right? So it doesn't buy you that much to have kind of 10 voxels across cortical depth if they all have the same value. For the function, there's a lot of information down there. And the kind of information that you can like infer from it for neuroscience application is something that I would like to show in this example, where I'm showing you two experiments. In both of them, the participants did a task. In one of the experiments, here at low resolutions, we asked the participants to do a tapping movement, kind of a pinch motion. Then for the other condition, we asked the participants not to move their fingers, but instead just to think about moving their fingers, do imaginary tapping, really focus on it. And you can see that, well, the activity like level, the strength might be slightly different, the shape of the blob is slightly different, but overall you cannot look at the brain pattern here and say, okay, this person is moving the fingers and this person isn't, right? Even though the behavior is fundamentally different. So when we zoom in to this tiny patch of the cortex and increase the resolution further and further and further until we are in the kind of sub-millimeter regime, we start to separate like these different sub-compartments of the blob, and namely these two stripes, which I believe refer to the different layer groups. For example, the superficial stripe over here is referring to layer 2-3, which are mostly responsible for cortical feedback input or general cortical input. For example, also input from motor planning areas or feedback from sensory areas and so on. Whereas the deeper stripe here is mostly referring to layer 5B, which have these huge Best cells, which have these huge axons down the spinal cord and trigger the muscles to move ultimately. And we see that both of them are engaged for tapping. Whereas for this imaginary tapping, we do not really see this secondary stripe, the deeper one. So suddenly we can kind of have a better understanding of the processes that then also match to the behavior, right? There's no output, no muscles are engaged here, even though there's a lot of activity going on, specifically in the superficial layers. Doing these layer fMRI experiments like these, however, taught me very early on that it's very hard to do layer-dependent fMRI without also at the same time doing kind of columnar imaging to, like, you really need to know where you are in the kind of topographical domain, which I would like to explain in this experimental series here, very similar to the previous one. Again, we asked the participants to do kind of tapping versus now not imaginary tapping, but just touching, which is slightly more robust. And we do get feedback into the superficial layers in the motor cortex. And in this patch here that is responsible for the pinch movement in the motor cortex, we again see this kind of bimodal pattern. We have the superficial activity. If you plot the activity, like brain response as a function of cortical depth, we have this input and we have the output down there. Whereas we only have the input for the touching without the output. However, and the point that I want to make here is that when you move just a few millimeters away, this kind of nice dissociation breaks down. And you don't see the difference anymore because you're not in the perfect column anymore that you care about. So you cannot really do like laminar imaging without knowing which part of the cortex here you care about. So we need kind of topographical acquisition and also analysis tools, which we had to develop. And again, here using the test bed of the primary motor system and looking at the individual finger representations. And I would like to show you how that works by looking at this little animation here, where we are looking at an axial like slice where the front of the brain would be right over there and the back of the brain would be right over there. And in the center, we have the central sulcus. And on the right side, you see the motor cortex. You see how thick it is, right? It's about 4 millimeters thick, as opposed to the sensory cortex here, which is not even 2 millimeters thick. It's one of the hardest areas with respect to like cortical layer dependent fMRI. The point I want to make here is that we can use kind of topographical patch flattening or using flooding algorithms to kind of, sorry, this is not what I wanted. I need to learn my shortcuts. Okay. Thanks. So I click on this one. Okay, Option, Shift. There we go. So now I'm kind of flooding the cortical ribbon in 3D in voxel space for the sensory side and the motor side in black and white, and then can kind of look at it in 3D. So now I'm looking at it from the perspective of the back of the brain, and everything in black is this somatosensory areas, and I'm projecting the individual fingers onto it. So this would be the representation of the pinky finger, the ring finger, the middle finger, index finger, and thumb. And you can see in the sensory cortex how nicely behaved that is, that you have these kind of fingers next to each other in the brain as they are next to each other on your hand, which kind of validates that the mapping tools kind of make sense at these high resolutions, but you don't really need high resolution to see that because you see how huge it is. Interestingly, then more on the motor side, when we flip this around and look at this little cave over here, which would be the hand knob, again, I'm projecting the individual fingers onto it. We have this pinky finger here. We have now 2 ring fingers. We have 2 middle fingers, 2 index fingers, and 2 thumbs. And not only that we have kind of these mirrored representations, but importantly also, they are tiny, right? So here the entire hand representation is as big as maybe 4 millimeters, which might be better appreciable when you look at it, a projection like that, where on the bottom left in the sensory cortex, we have one huge hand representation, homunculus style, whereas in the motor cortex, we have these kind of mirrored representations, which is also quite consistent across participants here, where we always have at least 2 mirror representations. And interestingly enough, the borderlines of these hand representations match quite nicely to patches in the motor cortex that are preferentially activated for kind of grasping versus retraction tasks, where again you can see the value of these high resolution studies, that kind of you see additional features and you learn that the motor cortex is not just a body part map that you might see at low spatial resolutions, but looking at it very fine, you see that it's actually more like an action map, as some monkey electrophysiology literature has suggested already. And it matters if you move your finger in that direction or that direction, right? Which muscle is being used. With all these kind of tools then being more and more established, excuse me, it didn't take very long until layer fMRI was also applied more in the kind of cognitive domains, like not only looking at primary motor and primary sensory areas, but also in these kind of associative areas. For example, DRPFC, which is a study by Emily Finn. It was one of the first ones looking outside the primary areas. It was very challenging with respect both to acquisition and analysis. But also in DRPFC, there are specific neuropopulations. For example, from the monkey electrophysiology, we know that there are these so-called cue cells and delay cells and response cells that ultimately trigger the motor system. And those different populations are located across different cortical layers. So Emily found an old task that should engage these different cells differently by showing participants these strings of letters like D, H, C, X, W, and then they are cued to either memorize the strings of letters in the order they saw them, or to alphabetize them mentally after they're already off the screen. Like, for example, to something like alphabetical orders of C, D, H, W, X, which is quite a hard task to do, and it's believed to mostly engage the superficial layers. Whereas later in the trials, then the participants need to do a response decision, which is then believed to mostly engage the deeper layers. And after a lot of kind of tweaking and really pushing the acquisition protocols for each participant's shift around the artifacts to be everywhere else, but in this tiny patch of the cortex, and also with a lot of kind of manual tweaking of selecting the areas and the signals from the right location, finally, we did see that the response condition really follows mostly the deeper cortical layers as opposed to the alphabetization, which is mostly following the activity in the superficial layers. Maybe better visible also in the time courses here of the group results, where you do see that the superficial layers, they care about this alphabetization, right? They shoot up as soon as you cue them to alphabetize, whereas the deeper layers don't do this so consistently and mostly shoot up as soon as participant found a solution and need to do a response decision. Though, as I kind of implied already, this area, this study was really focused on this patch of the cortex, right? Which is kind of ironic that like layer fMRI is a technology that promises and is kind of pushed for the purpose of addressing and answering new questions of directional functional connectivity, right? For example, we see which layer sends output, but we don't really see the connection, right? We only see input versus output, but we don't see the area where this output arrives, right? So it's funny to have a connectivity technology that doesn't really like show us the connections until that point. So there was a lot of move forwards to kind of larger coverages and to not only see brain systems, but also to look at connections across brain systems, across the entire brain, to not only look at these small patches of the cortex, like the motor cortex here, but to something where you can actually use the entire slice and then basically stack it together to end up with something like whole brain. And there are a few initiatives going in this direction. All of them benefit a lot from advanced EPI sampling schemes. And we also kind of benefited a bit on a specific T1 contrast generation technologies there. And one of the studies that I want to show you is the one from Kenju Koiso, where he tried to explore how much we can do layer-dependent fMRI at these whole brain protocols, and specifically he was looking into movie watching tasks. So there's a collection of movie clips from the HCP protocol. It's about 15 minutes and a collection of maybe 5 or 6 movie clips, and he did the studies 51 times spread across 13 sessions, 10 of them with fMRI. And you can see that the kind of movie networks are actually quite consistent across the sessions. Maybe session 6 is a bit weaker, but aside of that, it's very consistent, which tells us, first of all, that from the sequence perspective, I think the methodology is getting there. And there's still artifacts and it still can be optimized, but it makes sense to move into this direction. And because it's movies, you are not kind of constrained to these simple kind of on-off designs or univariate analyses, but actually you can push it further to pattern analyses. Specifically, we're looking at representational similarity analyses, where basically for each movie frame, you look at the fMRI signal pattern at high resolutions and see how similar this activation pattern is to all the other movie frames, giving you this kind of dissimilarity matrix, where if you squint your eyes a bit, might be able to see a bit of structure there already. For example, that some movie frames are just more similar than for example, others. And this might be better visible also looking at the averages within the movie clips, where you can see that some movie clips are just more similar and some movie clips are just super differently represented in the brain. And the interesting part with these kind of paradigms is that you can also directly look at it and compare it to behavior. So for example, just asking the person to rank which movie clips are more similar than others. For example, asking, are those 2 movie clips more similar or are those 2 movie clips more similar? And then you rank it up and you get a similar dissimilarity matrix, where again, you can see that in some areas, for example, here in the visual cortex, some movies are just perceived as they are represented more dissimilar as others. And interestingly enough, this is specific to some brain areas. Other, for example, here some frontal areas do not show this nice correspondence to behavior. And then you can look at the layers and see that the areas that show the correspondence with behavior are the ones that seem to be more middle layer driven, where the average similarity is mostly coming from the middle layers, as opposed to these other brain areas that do not match the behavior, which are kind of more feedback driven. And maybe I need to get a step back here and talk about feed forward and feedback. And actually, I have the next slide about that. So hang on there. So maybe you know, if you're in the field of neuroscience, that there are brain areas that are strictly hierarchically organized. And then based on like Velleman van Essen models, we know that each brain area in these hierarchical systems receive feed-forward input from another area that is lower in the hierarchy via bottom-up feed-forward input to the middle layers mostly. Whereas the same brain area receives feedback from another area that is higher in the hierarchy, mostly via top-down input, mostly in the upper and deeper layers. So based on this kind of canonical microcircuit, you can then build hypotheses about layer-dependent connections across large brain systems and networks. For example, if you just look at the functional connectivity of a given brain area A and look at the functional connection of each layer to all the layers in another brain area B, and you have these off-diagonal elements, it suggests that area A sends feed-forward input to area B. Whereas if area A sends receives feedback input from area B, it would more look like that. Or if both areas receive common feed-forward input, it would look like something like that. Or the connectivity matrix might be more feedback driven on both areas with common feedback, then it might look something like this. And you can also build null hypotheses, for example, suggesting that they might be connected the areas, but there is no layer dependent signature layer. It's just flat, or that layer dependent patterns are mostly explainable by vascular activity, which just tends to be stronger in the surface compared to the deeper layers, or just the noise level, which tends to be stronger in the surface. And then you can look at the empirical data and across, for example, here the visual system, and just see which kind of superposition of hypotheses best fit the data at the end. And then you can sort the areas or validate your kind of the Fellman-Van Ness model there, which is not only possible in the visual cortex, but also, for example, in the somatosensory areas. It's also believed that they are quite strictly kind of hierarchically organized, which I would like to show in this study here by Yinghua Yu and Jia Yang, who were looking at S1 specifically in the context of predictive coding. So also in the primary sensory areas, we do believe that most of the feed-forward input comes into the middle layers. And with feed-forward input, I mean kind of when you have a touch happening at your fingers, the input comes through the thalamus and enters the cortex in the primary sensory areas in the middle layers. Whereas other feedback input, cortical input, for example, with respect to arousal, attention, expectation, and so on, is mostly coming from superficial or deeper layers. And then we try to engage these different circuits differently by giving different touching tasks. So here, this is a 3D printed device. Inside the scanner is kind of a piano key mechanism where Yinghua was at the border of the scanner board and touched the participants in a specific order. And the participants learned to be touched, for example, here in the order of pinky finger, ring finger, middle finger, pinky finger, ring finger, middle finger, and index finger. In some instances, however, she left out the index finger, right? So there was no actual feed-forward input coming in. However, the participants, they expected to be touched on the index finger. So there was this expectation, the blue circuit, but there was not actual feed-forward input coming in. And in one of the few control conditions, then she also touched the participants in a completely random fashion, where they could not predict which finger is going to be touched next, which is then basically mostly engaged the green circuits, the feed-forward input, which you can then also see in the group results here for the layer profiles, where you see the fMRI response across cortical depth, where for the random touching, it's mostly kind of an inverted U shape. So the middle layers seem to be dominating the overall ongoing activity with a bit of deeper layers too here. Whereas for the prediction without actual feed-forward coming in, it's mostly dominated from the feedback superficial layers. And in a follow-up study just last year, they also used a variation of this design, which also maybe was a bit cleaner to also separate just the effect of surprise to kind of be touched without expecting to be touched, but from the actual prediction error from the expectation versus the surprise in that sense, where they again could reproduce the modulation in the superficial layers. However, having a rhythmic design and then having a kind of mismatched rhythmic delay, they could also modulate on top of that the activity in the deeper layers, the feedback in the deeper layers. These principles of layer-dependent fMRI can also be applied in the clinical context. So here, this is a collaboration with Silvina Horowitz, where we try to see if layer-dependent fMRI can inform neurology, specifically looking at focal hand dystonia patients. And these are patients that kind of are like almost like regular people, but for some tasks, they just cannot like do the movement. Like, for example, for writing, the hand just cramps, right? Then in this case, it's called writer's cramps. And it's actually quite one of the most common neurological disorders. Musicians, it's known too that you can like, do your daily work, but then for playing the instrument, you cramp. And there are a lot of theories, or some theories, competing theories about where this comes from. And one of the theories is that it might be due to a lack or reduced surround inhibition. So with layer-dependent fMRI, we might be able to have some, provide some information in this discussion, because we do approach these kind of microcircuits there. But first of all, here for a finger tapping task across 4 patients, we can see that there is actually quite a lot of activity. So despite all the challenges in the clinical context, you can get like good data quality. And then when we look at these kind of topographical mappings here with a specific program that's in Laney from Faruk Gulban, and I will talk about that in a few slides, and namely the patch flattening, you can look at the primary somatosensory cortex and look at the individual finger representations here. And for the unaffected hemisphere, you do see that the fingers are right next to each other as expected. For the affected hemisphere, however, you see that there's a lot going on. We have a lot of significant activity. However, there's not this clear kind of structure that each finger is right next to each other. There's a lot of overlap. It's actually, we wanted to kind of quantify the overlap and the location, but you see how messy it is, right? How do you quantify this? It's just all over the place. More quantifiable, maybe, when you look at the layers on the motor side of the central sulcus, we do see consistently that the affected hemispheres have this preference of the superficial layers as opposed to the deeper layers, which are just weaker. As opposed to the unaffected hemispheres, where again we have this kind of input-output characteristic of the motor cortex, this double bump, and these bumps are just modulated for the 2 different hemispheres, for the affected and unaffected ones, which nicely goes along with the idea of the surround inhibition that's lacking or affected and reduced in the hand dystonia patients. So quite exciting. And so I think layer-dependent fMRI can have value also in these clinical contexts. Though I think until now, most of the applications are from layer-dependent fMRI are more in the neuroscience domain. And we're also not quite there yet to make it really kind of turnkey neuroscience methods. And for example, one of the limitations so far has been kind of the temporal constraints of common neuroscience tasks, which is why I want to also show this study from Sebastian Dresbach, who is trying to explore the usability of layer-dependent fMRI for event-related designs, which is a big deal in neuroscience studies. So we really pushed the sequence, the acquisition procedure to be quite fast, and then look at kind of event-related designs and block designs for visual and somatosensory stimulation. And I guess the first thing that you clearly see is that when you push the sequence to be so fast, you just pay a huge price in your stability. You need, like, you cannot be fast without compromises. So you have a higher noise level. Of course, you have more data points too, but the efficiency, that the number of increase of data points that you have can never account for the loss in sensitivity that you get in the first place. On top of that, we also see that the response magnitude is just weaker for event-related designs as opposed to block designs. So it's kind of a double blow there. So you just need to scan at least like 4 times, 8 times longer with event-related designs to have the same kind of activation statistic significant scores that you would have for a slow design with long TRs. Importantly, however, when you average long enough, and then you do see these kind of faint modulations in onset timing and the HRFs, for example, that the deep layers are just faster than the superficial layers, which I think has quite nice implications for the usability to look at also the temporal dynamics of layer fMRI processing. And of course here, this is the reason why we see superficial layers slightly deeper, slightly delayed, could be partly due to kind of the different hemodynamic response functions across cortical depth or the different neuroscience processing. But now we know that we can kind of modulate these timings, for example, with different tasks, and we do have the ability to see those. Also, in order to make layer-dependent fMRI more usable for neuroscience, I think we need to move across field strength. And this, there are many motivations to go there, in the sense that, like, not everybody is at one of those kind of prestigious research institutes that have access to a 7 Tesla. So to be more kind of accessible, we need to go to 3 Tesla. And the field of layer-dependent fMRI is just growing a lot these days. And the ultra-high field community is growing too, but not with the rates of layer-dependent fMRI. So to meet the demand of users, we need to move across field strength. And also from a sequence developer perspective, 3 Tesla is awesome because you have like orders of magnitudes more users, right, that will cite your papers and use your tools. But also, it's just easier, right? So here in this group study with Martin Kronbichler from Salzburg, you can see the underlays, which is the mean EPI. It just looks like an anatomical scan, right? Like to do EPI at 3 Tesla is just way easier because you don't have all these B0 problems. You don't have these B1 issues. The noise level is higher though. So you need to scan a bit longer to see kind of that activity of here, for example, with the unilateral tapping task in the motor cortex. But then if you scan for, I think, 24 minutes, you do get the indications of the double bump feature in the motor cortex and also how the superficial bump is modulated for kind of looking at the ipsilateral tapping versus the contralateral tapping. So in Maastricht, where I was before I moved to US, we had different field strengths available. So I tried layer-dependent fMRI vaso blood volume imaging across field strength. And I do think that you can make it happen across field strength. It just works. You have different challenges as different field strength. So at 9.4 Tesla, it's more an engineering challenge. You need to kind of have kind of a whole team of engineers helping you. And then you have the best SNR that you might wish for at a lot of other, like, but to get there, it's challenging. 7 Tesla is way more turnkey, and 3 Tesla is just push button, right? And then you get layers out of it, but you need to scan longer compared to 7 Tesla. So 7 Tesla is a nice sweet spot there, I think. Also with respect to cost efficiency, because it's still more cost efficient to have shorter experiments at 7 Tesla as opposed to like very long scanning at 3 Tesla. So aside of all these acquisition developments and challenges that I talked about so far, I think there's also the challenge of the analysis. And because layer-dependent fMRI does have absurd high quality demands, so you really need to know if your voxel refers to this layer or that layer. However, also layer-dependent fMRI are messy, right? The data are all over the place and there are artifacts. So common analysis softwares are not really kind of able to meet this weird, like having high demands, but giving it bad data compromise. So I think we need a kind of layer fMRI optimized software suite, which I'm trying to develop. And the software is called Laney. And Farouk Gulban joined in the first wave of COVID and has nicely reworked, professionalized, and increased the capability of this software suite quite a bit. And the kind of demands that this software is trying to meet is, for example, to kind of just be optimized for the usual data that layer fMRI folks are acquiring. For example, layer fMRI has a lot of voxels, so it takes a lot of time to acquire it. And it sometimes can be wasteful to do a whole brain acquisition if you have only like specific hypotheses about a subset of brain areas. So most people do slab-wise acquisition, but slabs do not fulfill the common topology requirements of standard mesh-based layering analyses like FreeSurfer or so. And very often you have these kind of unconnected spots. You just definitely don't have closed surfaces without holes. Then also in layer-dependent fMRI, we have the thing that I call the beer-wine dilemma. So while conventional fMRI, representing the beer here, has been standardized over decades to give you data that are homogeneously pushable through standard pipelines, like done by multinational brands of beer corporation, layer fMRI, representing the wine here, is way more specialized, right? You have like specific data and study specific protocols and challenges, you need more flexibility and quality control, especially in these kind of early days of the field. So like wine, in layer-dependent fMRI, you do get the best results, not by kind of homogenizing the processing in a Henry Ford assembly line production, but rather to allow more flexibility and adjust your software needs based on the specific study, like you would for wine, right? Based on the country you're in or the season or whatever. It's not a factory, like standardized factory as it is for beer, which however comes along with other challenges. For example, the need for standardization in like neuroimaging communities. So we need to find a compromise between kind of flexibility of the analysis without really allowing too much kind of interventions and introducing biases and data manipulation. Also in layer-dependent fMRI, it's quite common to use contrasts that are not only gradient echo bold, but also other contrasts like VASO or vapor or ASL or you name it. So we need analysis tools that deal with that too. And also importantly, as I alluded to that already a lot, layer fMRI has issues. We are bandwidth limited. So we have like ghosting artifacts more than conventional fMRI. We have a lot of noise level. So we need tools to deal with that too. And the Laney program that is, I think, the most popular one is just the layerification. So when you give Laney a kind of segmentation of gray matter, white matter, and CSF tissue types, you need to kind of have tools that tell you which voxel refers to which layer. And this is an example here for validation purposes, ex vivo in the primary visual cortex, where you do see that the stria genari kind of over amplifies the curvature. So the stria genari is very, very close to CSF as opposed to white matter borders at the crown of a gyrus, whereas at the bottom of a sulcus, it's very, very close to white matter, right? So if you just take the middle layer, the so-called equidistant layering, you kind of miss large parts of the stria genari. So we need these kind of equivolume approaches, which is based on and implemented directly in voxel space in Laney here. And you can validate this also on kind of ex vivo histology data, for example, the 3D whole brain 100 micrometer and big brain. Also, another core program in Laney is the generation of columns by just iteratively dividing the 3D voxel ribbon, then allowing the user to kind of unfold and flatten the cortex without interpolation to vertices, but directly as a kind of voxel intensity remapping, which I showed you earlier already with respect to these mirrored finger representations. But also in cases where you can assume that neighboring columns have similar layer activity, layer-specific smoothing without kind of signal leakage across layers can be very helpful to deal with the noise level of individual runs. So it can look quite like frustrating, but if you know that neighboring columns do the same things and just do smoothing without signal leakage across steps, then you can also use layer smoothing to explore the data like that. All right, so to close the story, I think layer fMRI can be informative for neuroscience specifically, because neuroscience already bridges a lot of different spatial scales, all the way from the microscopic side, where we have the synapses and the neurons, the cell bodies, all the way up to the macroscopic side, where we have behavior, manifestations of mental disorders, or the different brain areas. And it's only this kind of mesoscopic part here that's somewhat underrepresented. But neurotechnology allows us to bridge the two sides further and further. For example, new optical methods allow us to capture hundreds and thousands of neurons at the same time, whereas high-resolution fMRI does not only map activity changes across brain areas, but also within brain areas. The keystone that will make it stable and allows us to really build on top of that until sky's the limit is the spatial scale of a few hundred micrometers, where we have these basic neural building blocks of the layers and the columns. So I think that's important to bridge the gaps there. I would also like to do a bit of advertisement. So if you are interested in layer-dependent fMRI, there is this interest group, the Laminauts at laminauts.com, and we organize layer dinners. Those are until now mostly kind of virtual conferences. The proceedings are all online, and we also have kind of more student-based initiatives. For example, right now we are setting up an analysis challenge or a survey, if you will, where we want to see how people do layer-dependent fMRI and then also focus our teaching efforts in that direction. Basically, just downloading an example data set of EPI and anatomy, and then showing how you get from that to statistically significant layer profile differences here. On Twitter, as layer-dependent fMRI, I try to keep on top of the field and mention and note anything that's happening in the field, including papers that come out, conferences or initiatives like that. Also on the blog of layer-dependent fMRI, I try to kind of collect all those kind of information that do not really find their way into the academic literature. For example, kind of how to hack your scanner, like, or specific Siemens scanners, for example. How do you optimize it that it's not done for clinical whole brain imaging, which they are being built for, but for high resolution layer dependent fMRI, also for analysis, right? There's a lot of kind of know-how, how you use ITK-SNAP or you name it to do layer dependent fMRI analysis, which is just not science, right? And therefore it never finds its way to the academic literature. On the blog, including some opinion pieces, I try to collect that. Also, there are two exciting workshops coming up with respect to layer-dependent fMRI at MGH in early October, mostly organized by Jonathan Polymany. There will be really an educational 4-day course where people will not show kind of these science lectures like mine just now, showing like fancy images, but really going like down to the kind of the principles. What are the basic steps? What are the assumptions and the common misassumptions in layer-dependent fMRI analysis or acquisition, for example, understanding the basics. And then there's also a 2-day hands-on course right before the Minnesota workshop, slightly later in October, where we will, organized by Luca Visioli, where we will mostly talk about kind of different approaches of do layer-dependent fMRI analyses, like with Brain Voyager, with Laney, or with FreeSurfer, and then basically showing how different groups do things. With that, I want to thank my colleagues. This is my new group at NIH. It's headed by Peter Bandettini. This is the core FMRIF. Before that, I was working with Benedict Posers, who contributed the sequence that was used in many of the data collections that I showed you today. On the new generation of scanners, that is the Terras, I'm using the sequence from DZNE in Bonn, specifically Rüdiger Stürnberg, which is just a fantastic sequence. And with this, I thank you for your attention.
If you have any questions, I ask you to unmute yourself and ask. And if someone's already asking, maybe raise a hand. We have about 15 minutes for questions. Stefan, please unmute yourself.
Well, thank you very much for that very nice and interesting presentation. I was wondering about the representation of the movements in the motor cortex. What I thought was that in the motor cortex, you have broad overlapping muscle representation with mostly complex movements represented more than just the fingers. How do you we reconcile that with what you have shown?
I think this is pretty much what we see. Like that, we see kind of distinct representations if you do kind of grasping versus retraction, and which is kind of one of those movements that they showed in monkeys, like these complex movements, the hand to mouth. So when you stimulate at kind of macroscopic time scales for a long time, then the monkeys do these kind of complex representations. I think, well, I should be specific. So we look specifically at the Brodmann area for A, right, the anterior part, so the evolutionary older part. And if you go down the central sulcus a bit further, it gets pretty messy, and I don't really see clear maps anymore. And I think there is this kind of, in the literature, there's this distinction between kind of the fMRI community, which basically treats the motor cortex as a homunculus, because I think just because in gradient echo bold, you have the big veins and you cannot really separate the sensor from motor cortex, even though people think they do. So in the fMRI community until now, almost like all the studies that I found basically treat the motor cortex as a body part map. And kind of with these high resolutions, we can, I think, kind of bridge the models that the electrophysiology sees, where they go more into the direction of kind of movement representations or muscle representations to that degree, where it matters if you move your fingers basically on with this muscle or on that muscle, extension or flexion, where we kind of see where at which spatial scale this breaks down, and where the fMRI literature kind of then matches better with the electrophysiology literature to some degree. If that answers your question. Yeah.
These maps are quite plastic. When you train a subject or a monkey, then you see a huge change in the overlap and the representation of these maps. Did you try to do that as well?
Well, we didn't modulate it. There are a lot of exciting studies trying to maybe go into that direction with kind of gluing fingers together for a week, and they already see how it remaps. I didn't, like, I looked at the specificity, for example, just along the cortical ribbon to see kind of what I showed in the maps was kind of the strongest activity, not really like a winner takes all, because it was kind of a gradual, but it kind of it was a selectivity map. But when you look at the individual digit response, you see that for just the index finger, most of the motor cortex becomes activated, and then just this one patch has kind of a slightly stronger activity compared to the other fingers. So there's a lot of overlap there. But we didn't do kind of plasticity studies for that. No, we were just, as I introduced it, it was more kind of developing the tools, having a playground to see topographical representations, and then this mirrored representation jumped onto it. And maybe there is this I would want to mention it in bioRxiv. There's this new paper from even Gordon, I think. It made a big splash a few months ago, which apparently is now accepted in Nature. So they show this also for kind of bigger brain structures, which are all kind of mirrored. Maybe you're aware, which I'm talking about. I can post it in the chat later, where I think they also tried to make the connection that in the motor cortex, kind of it is aligned along the central sulcus if you refer to the different spines, right? The one spine is referred to the next spine, but if you look at the body parts within a spine, then it gets kind of messy, which is, I think, what you suggest and what I see too. Yeah, we don't look at plasticity. No,
great.
Yeah.
We have a question from Jean-Luc in the chat. Between 70 and 3T, by what percentage would the protocol need to be lengthened to achieve the same level of significance and spatial resolution?
Sorry, it is 3T 70. What would need to change? Well, it depends on your question, I guess. For not much, I think. Usually we adjust the protocol to push it as much as the hardware allows. And at 3 Tesla, we usually have kind of 64 channel coils or 32 channel coils, which is kind of getting the standard, or is the standard at 7 Tesla, the 32 channel coil. So Grappa would be just as much as possible. And also with these large matrix sizes that we have for layer-dependent fMRI, we just want to acquire it as fast as possible. And usually we have echo times that are on the longer end anyway. So, and the body gradients or the Prisma, for example, that people would use for efficient fMRI are identical compared to 7 Tesla on the Terra or the Magnetome for that matter. I'm not sure which, I think you have the Terra, right? I have a collection of protocols on GitHub at layerfMRI/sequence git, I think. And there you can see that it's almost the same protocol. The one thing that you might want to adjust for VASO, which is a method that I use a lot, blood volume sensitive, is that you have an additional inversion pulse for every TR to get a kind of blood volume sensitivity, and not only kind of the susceptibility that people do in BOLD. There you would usually have slightly shorter inversion delays, which makes 3 Tesla slightly more efficient. But usually, yeah. That was the best.
Thank you very much. Just for precising, I was thinking about the time of the experiment itself. You said that we have to scan longer with 3T than 70, and perhaps I missed it, but I was wondering how long longer we should for getting the same significativity and spatial resolution in terms of signal.
I think it's hard to really quantify. I can tell you about my experience that I like in 7 Tesla for a finger tapping task, which is a huge task, it takes about 12 minutes to get a decent looking map. So if you really want to have a map where you have an activity score for each voxel, very often you don't want that, and you want just a layer profiles and can average across a chunk of the brain, then you can be faster. At 3 Tesla, I need at least 3 times as much. So the different setup of your experimental protocol would be a factor of 3, in my experience, that I need about 45 minutes at 3 Tesla for something that I get maybe in 15, 12 minutes at 7 Tesla. And in Maastricht, the hourly rate that you pay for using the scanner was proportional to the field strength. So at 7 Tesla, it's 2.3 times more expensive than 3 Tesla. And therefore, I said at 7 Tesla is more cost efficient, because even though at 3 Tesla, it's cheaper to measure, it's not a factor of 3 cheaper, not just yet, right? So therefore 7 Tesla is more efficient.
Okay.
But this is like, it's hard. It has a different RF coil, right? Which one is built by Siemens, the other one is built by Nova. Yeah, and in the 3 Tesla, it's kind of a new field, right? 7 Tesla, I've optimized this for and over-engineered my protocols for a decade at least, right? At 3 Tesla, we just went there and tried it, and it kind of worked. And you need 3 times. So it's not a physical, like a scientific comparison. This is my experience. Yeah.
Okay, thank you very much. Thank you.
Cécile, you can unmute and ask your question.
Hi, thank you very much for this wonderful talk. I have just 2 questions, one very quick, and the second one maybe requires more discussion. The first one, you talked about at one point the whole brain layer fMRI. And so I was wondering, it's similarly to the question of Jean-Luc, how long it takes to do a whole brain layer fMRI?
Yeah, longer than a slab for sure. It's wasteful. Like, you acquire a lot of stuff and a lot of brain areas, and it only makes sense if you really need all the brain areas. So it depends. So kind of the map that I showed with this sessions sensitivity, maybe I go back to that. And in the meantime, for Stephanie, I will post a link to that paper that I was referring to, which caused so much excitement about the motor cortex in fMRI.
Thank you.
It is, I think, partly a strawman story, and because, as you implied, and as if you look at Penfield originally, the original literature, that you know that, like, people know that there's a lot of overlap, that the motor cortex is not a homunculus, and they can anyway. Sorry, back to your question. How long does it take to get to whole brain? Where is this slide? And I need to share. And you see this. I will change presenter mode. Can I move you? There. So this is the strongest. I think it's something like a visual attention network. I have a kind of DRPFC in there. I have interparental sulcus in there, which is also the zoomed section. And this was 5 averages that we ran per session. So this refers to 75 minutes. Then I get networks at this kind of quality.
Okay.
So if you are after kind of maps like this, then you need more than an hour. If you do, maybe I have this somewhere else. If you just do kind of a flickering checkerboard with finger tapping, then you get something, I think, in 45 minutes.
I see. Thank you very much.
Sure.
Yeah. And my second question, because I was a little bit biased with your presentation with the work with Silvina. I was in the lab of Mark just before. I came to the Paris Brain Institute, and I'm very interested in surround inhibition because I'm still working in the field of dystonia. So I was wondering how you could, because you talked about it, but in the results you presented, there is not really a link between the results and surround inhibition. So what would be your way of testing it? Is it possible to do an inter-layer directed connectivity, or because in the surround inhibition, actually the inhibition should come from the representation of the neighboring fingers. And if you have multiple neighboring representations, it's very difficult to test this idea of surround inhibition. So I just wanted to have your idea about that.
Right. So this, like, the goal of that study was more to kind of to see if we see something, and then we saw something. And the exciting thing there was we didn't only see activity, despite all the challenges that you have in the limited like scan time of patients, but also that there are already kind of indications that the microcircuitry is different. The layer profiles did not look identical. So the surround inhibition, in my naive understanding, is that it's believed to be mostly in kind of in the feedback layers, like in the motor cortex, the superficial layers, that the kind of the lateral connections would be stronger, or the lateral inhibition would be stronger in the superficial layers compared to the kind of the output layers. And because we see that we see the kind of the response change in the right direction for patients, namely that the superficial layers are way stronger than the deeper layers in these affected hemispheres, which could mean that the superficial layers need just more work, or that the superficial layers are just not as suppressed from the surround as the deeper layers, where we wouldn't really expect this surround inhibition as strongly, I believe, in the kind of output layers. So that was kind of my clue. With Sebastian Driesbach, who has showed results from in the visual cortex with event-related designs, he's actually exploring kind of a local connectivity. So to look at the kind of one finger representation, the somatosensory areas, and then the next finger representation, and the next finger representation, looking at kind of lateral connectivity, functional connectivity for that matter, which might go more into the direction what you're proposing, though. Kind of there are a lot of kind of other issues too. For example, the vasculature, for example, the diving veins, they have much larger branch sizes in the deeper layers as opposed to the superficial layers. So it's kind of still quite hard to really disentangle the kind of the neural driven signal from the kind of vascular artifacts in resting state connectivity, for that matter, which, So as soon as you go to kind of resting state connectivity, then, or you don't have a ground truth of your neural fluctuation, and you look at the neural fluctuation, like correlation between these different patches or finger representation, it's also becoming very hard real quick. So I don't know the answer for you, I guess.
Okay.
There are the different directions, I guess.
Yeah. Thank you very much.
Sure. You have been our editor, wasn't it, for that paper? Thanks, yeah.
Any more questions? Okay, I have one. So you mentioned the working memory study in the dorsolateral prefrontal cortex, right? So that study essentially was looking at the part of either alphabetizing or the response time, which is in a way an event design, and also in an association cortex. So I believe maybe for working memory, there's not much adaptation happening. So I imagine, I'm not the expert for working memory, the response in the dorsolateral prefrontal cortex would be stronger. But then just to get an idea, how many trials for this kind of a study, and what would be the intertrial interval that you were looking at for an association cortex?
Maybe I need to pull it up again. That was quite early. So maybe that the lessons I learned on that study might not be kind of so valid now, but I can tell you. Basic share. Oh, not this one. So here overall we had intertrial intervals of 16 seconds, and we had kind of the alphabetizing was 9 seconds. And Emily, she's a neuroscientist, she really wanted it faster. And it was kind of, this was our compromise, because I wanted to push it way slower, but she said it wouldn't be natural anymore. Maybe you know what that means. I'm not sure. I know, like if you kind of keep it in your working memory too long. So the issue, the worries that I had was that the HRF is different across layers. We know that kind of superficial layers have a different composition of big vessels and small vessels, and we do know that big vessels and small vessels have different kind of vascular response functions. So However, as soon as they plateau, then you're not as sensitive to that effect anymore. So I wanted it very long. Also, the interstimulus interval was 16 seconds, which is kind of fast for my usual experiments, but it's kind of slow for many neuroscience experiments, because I wanted to really have the time for the HRF to go down and not kind of have a different steady state in superficial layers compared to deeper layers. Now people did more dynamic studies too. For example, for the imaginary TAPI, Andrew Persichetti, they looked also at kind of saturation effects and if you at the time courses of those, and they are getting very promising results there, or published this in Current Biology, I think. So maybe I'm too conservative, but it's hard. So for those study, this was the kind of the first one where I ever used an entire slice, and the artifacts were horrible, and there was a lot of tweaking necessary on the scanner. So we put a person in, looked at the data, yeah, it looked horrible. Then we tweaked everything, and then for another person, we had to tweak it very differently because the structure was very different. We had to do this online localizer. So this was at a stage where the sequences were not as ready as they are yet. So I think nowadays, with more validation of these, that you can actually do fast imaging and also with more stable sequences, I think it might be fine to go down to 6 seconds or something like that. Still, it might be cleaner to have it as long as possible to avoid differences in the HRF. However, there are clever designs in neuroscience where you then not only look at the kind of habituation, but also then the differences of differences. So basically that the HRF bias that you might have across different layers is basically subtract out. So does this answer your question? I'm not sure. It was quite slow for common.
Okay, because I think you probably know there was this discussion with you that we want to implement WASO at our Center for Motor Adaptation task. And so, yeah, I think I wrote an email to you as well, but maybe we can continue the discussion some other time or in the next round of discussion. So if the general audience does not have any more questions, maybe the students who registered for the tutored seminar can stay, and the others are free to disconnect if you don't have questions.